So, hello everyone, my name is Carlos Marti, and I'm a postdoc with David McCandless at Cold Spring Harbor, where we work on computational methods to predict how multiple mutations combine. So we've heard a bunch of talks of people doing this with neural networks, and I'm going to present these alternative methods that are called Gaussian processes, which have two main advantages, which are that they are more interpretable and that they are better at quantifying uncertainty. So I'll first give you a small introduction about how these models work and how we can use them for modeling sequence function relationships. I'll explain you new models that we've been developing that allow us to get maps like this that show how different mutations affect the predictability of other mutations. And then I'll show you how we've been applying these methods in a bunch of different data sets and finish with the conclusions. So I don't have to convince anyone here of the great impact that these high-throughput phenotyping experiments have had on our understanding of basic biology, on the function of different proteins, and on the consequences of genetic variation on human diseases. But one of the nice things that we can also do is we can start studying how the effects of multiple mutations combine together. In some cases, this is a complicated problem because the effect of mutations can change depending on the genetic background in which they are introduced. In some cases, this can be moderately simple, where we see that the effect of the combination of mutations can be predicted as a function of the effects of the single-point mutations. And this is tightly related to biophysical models of sequence function relationships, where we see that mutations affect biophysical traits, such as the binding energy of a drug to a sequence in particular configurations. But in many cases, this is a bit more complicated, and the effects of mutations really depend specifically on the sequence in which we are introducing them. So we typically think about this problem from this perspective of the genotype-phenotype map, where we just assign a phenotype to every possible genotype in the space. Here, I'm showing you a small example with two alleles and two sites, and we have this vector that represents the complete genotype-phenotype map. So the way these models work is we place a prior distribution over the set of possible genotype-phenotype maps that allows us to give a probability value to any genotype-phenotype map. And we can, for instance, say that this alternative genotype-phenotype map is less likely under the prior. We can also draw samples from the prior distribution, and we can draw as many as we want to kind of see the behavior that this prior has. But the really nice thing is that once we get measurements for a few of these genotypes, we can compute the posterior distribution, which is in some way we take the samples from the prior distribution that are compatible with the data. And we end up with this distribution, with this posterior distribution that give us an idea of what the possible genotype-phenotype maps could be. And we can ask any question we want about it. And the nice thing is that this is also a multivariate Gaussian distribution where we can compute analytically the mean and the covariance matrix. The challenge here is that this computation is very hard when we have a lot of data points. Right, so in the past, we've been dealing with this problem by using a bunch of mathematical trees that allow us to do this computation for millions of genotypes whenever we can enumerate all possible sequences in the space. So these tools are implemented in this Python package that is very originally called GPMap tools. And I talked about this in the variant effects seminar series. We have a preprint. And we have used these tools to now analyze a bunch of different complete combinatorial data sets, including regulatory sequences, protein-protein interactions. And we've also used it to see what's the impact of the structure of the genetic code has on these maps. But these are really limited to cases where we can enumerate all possible sequences. But thanks to recent developments in the Gaussian process literature, we can now do these computations in any case without restriction on the size of sequence space. So we can start doing this very generally in every data set that we get our hands on. So even though we can really reconstruct all possible genotypes, what we can do is the same way we are doing when experiments. We can at least make phenotypic predictions of unobserved genotypes. We can predict the effects of mutational effects in different genetic backgrounds, and we can also reconstruct small parts of this genotype-phenotype map. But the nice thing is that we can all do this while rigorously quantifying uncertainty. So now that I explained what are the main advantages of using these models for the modeling sequence function relationships, I'll tell you about the new models that we've been working with. So something I haven't told you much about yet is how do we define these prior distributions on genotype-phenotype maps. So we typically do it by thinking about the predictability of mutational effects across genetic backgrounds that differ at a number of positions. So here we have the same mutation that in genetic background then differ 1 position, here by 2 positions, and so on and so forth. So in the past, we developed these prior distributions that assign probability depending on the average squared epistatic coefficient across all possible pairs of mutations. We can also define priors that are parameterized by the variance associated to genetic interactions of every possible order. And the nice thing is that we can learn the parameters of these priors from this statistic from the data that we call the distance correlation function that tells us how much 2 sequences are correlated depending on the Hamming distance that separates them. This is showing you an example from this data set in which they measured the splicing efficiency of every possible 5' splice site. But really the data has more information than this. We can stratify these correlations now depending on which sites are actually different between every pair of mutations. And we can see that some sites have only a moderate effect on phenotypic predictability, but other sites, when we mutate them, they have very strong effect on predictability. And we can see this for every possible site in this genotype-phenotype map to see the whole range of variation that we can get. So based on this observation, we developed these new models that can account for these heterogeneous effects of mutations on the predictability. And in this case, we came up with this idea in which we have what we call decay factors that tell us the % decrease in predictability when we make a mutation. And they accumulate the same way the discounts in the supermarket accumulate. So first you get a 50% discount, and then you get like a 20% discount. And in this case, every mutation within a single site has the same effect on predictability, but we can have allele-specific effects or even mutation-specific effects. So we would have one of these heat maps for every position. And the nice thing is that we can infer all these parameters from the data and then use these parameters to guide the exploration of these complex genotype-phenotype maps. So that's what we're going to do now in these 2 data sets. I'll start first with this amazing data set generated in Michael Desai's lab, in which they took these 2 GS wild-type strains, they crossed them and let them recombine, so they have a lot of combinations, and then they measured the growth rate of all these segregating variants in multiple conditions. I'm going to show you only the growth rates in high lithium conditions today. Here I'm showing you the predictive performance in held-out data for different models using different amounts of training data. And what we can see is our new model performs much better than the previous models on any amount of training data. But the best thing really is that now we can make these maps. This looks like a Manhattan plot, but now on the y-axis we have this decay factor that tells us how much each mutations at each of these locus affect the predictability of other mutations. And we can clearly see that there is a single site that has a very large effect of about 45%, and this corresponds to this ENA1 gene. So we can look at the data, stratify it by the allele at this position, and we can see that this has a very large effect on the fitness. This is not surprising because ENA1 is the sodium-lithium pump and is the main mechanism of detoxification of lithium. So if we have a variant that doesn't work as well, then the growth rate is lower. However, we still see that there is a lot of phenotypic variation within each allelic background, right? So the next thing we did is we looked back at this map and took the sites that have the largest decay factors to make phenotypic predictions using the Gaussian process models for all possible allelic combinations in the 2 wild-type backgrounds. So this is showing over 100,000 genotypes. And they are arranged in a way that they depend on the Hamming distance, and they are colored by the allele at the ENA1 locus. So what this is showing you is that the mutational effects depend really a lot in the allele at the ENA1 locus. And not only that, we can, even in the bad version, there is a bunch of genotypic combinations that give a variant that is even fitter than the 2 wild-type strains. So there is place for a lot of compensation, even when you have a bad version of this variant. But what we can do next is, this is one of my favorite plots here, is that we can make predictions of the effects of mutations in genetic backgrounds that differ only at these 2 positions. And this is really cool because the data that we get are only random recombinants, so we actually never measure the effects of these mutations in these specific backgrounds. But still, you can see that we can say with a high confidence that the effects of some of these mutations are very different in these 2 backgrounds. To show you just one example, we have this mutation associated to the HAL9 gene, which is neutral in this BY background, which is the functional one, but is very beneficial in the RM deficient background. And this is also not surprising because HAL9 is a direct regulator of ENA1. So so whenever we have a bad copy of the ENA1, it probably acts by overexpressing the protein and partially compensating for the loss of function. So in the second example, we'll be looking at the capsid protein of the AAV2 virus in this data set that we took from protein gene that was generated at George Church's lab, in which they heavily mutagenized this 28 amino acid region in the capsid protein and measured the efficiency with which this virus assembles. So as before, we compared our new models with previous models, and we can see that the new models perform better given sufficient amount of training data. But the really cool thing is that we can get now these maps, which instead of showing the effects of mutations, now what these maps are showing is how much mutations involving specific alleles affect the predictability of other mutations. So you can see that this is a pretty heterogeneous map, and it's not, every position behaves very differently. So one of the simplest things we can do first is we can look at the allele with the largest decay factor. And this happens to be this asparagine at position 569. When we look at the data, we see that this is required for functionality. So whenever we have this allele, it doesn't matter which other alleles we have at other positions. And this asparagine is located in a very varied region of the structure and is forming this dense network of hydrogen bonds. So whenever we introduce a mutation, even to the most conservative amino acid, it either breaks the hydrogen bonds or induces steric clashes. So it cannot be replaced by anything here, basically. A second example is we find here this region that has systematically high decay factors for lysine and arginine. And this region is located in the surface of the capsid protein, and it's not very clear how it functions or what the requirements are there. So another nice thing that we do with the Gaussian process models, we can now make these predictions of single point mutations to create these heat maps that everyone is used to seeing here. And what we can see here is that there seems to be a lot of beneficial mutations, and most of them are actually the ones that decrease the charge in this region. So we can use the Gaussian process models to see, okay, let's see how these effects of mutations change if we change the charge. So we can add these additional mutations, and now we can see that many of these effects become neutral. And if we add a second mutation that decreases the charge, some of these effects now become deleterious. So we think that the logic of genetic interactions in these structures can actually be explained by this simple pattern in which if we stratify the data based on the charge at this region, we can see that an intermediate level of charge is required for functionality. So you cannot have too much positive charge or too much negative charge. So finally, we can look at these maps where we can see the effects that specific mutations have on the predictability of other mutations. And this is really nice because you can see that all these maps are very different. And this is highlighting that amino acid exchangeability can be very different across multiple positions, and the biochemical requirements depend a lot on the context in which they are introduced. Just to show you one example of how this works, we have position 576, where we have a very clear pattern with 2 blocks of amino acids. So whenever this is white, it means that when we make a mutation, the predictability of other mutation doesn't change, so we can make any mutation in this cluster or in this cluster, and we would basically have the same effects of mutations, but if we make a mutation going from one cluster to another, then the predictability changes. And these clusters are defined based on whether they are an aromatic residue or not. Again, we can see that the aromatic residues are required for functionality here. And when we look at the structure, we can see that the wild-type tyrosine is filling this hydrophobic pocket at the interface between 2 monomers. So it can be replaced by other aromatic residues, but not by any other amino acid. So finally, just to sum up what I've told you today, I've shown you this interesting class of models that are called Gaussian process models that we can use for modeling sequence function relationships that have these very cool properties. I've shown you new models that allow us to infer these site, allele, and mutation-specific decay factors, which are very useful to guide the exploration of these complex genotype-phenotype maps. And when we can use them to start predicting the effects of mutations across multiple genetic contexts. We can reconstruct small pieces of the genotype-phenotype map to see how multiple mutations interact with each other. And one of the things that I'll keep emphasizing is that we can do this while rigorously quantifying uncertainty, which is very important for knowing the evidence that we have for our conclusions. So moving forward, one of the things that I'm very interested in is in further developing these models to now account for interactions with the environment or different measurements. So once we have a library, we can make measurements in multiple conditions, and the effects of mutations may also change there. So finally, I would like to finish by acknowledging everyone in the McCandless lab, especially David for his guidance throughout these projects and other projects we've been working together throughout these years. And also, I want to thank Anna, Joanna, and Sam, who have been very instrumental in this work and have developed most of the theoretical results that are in this project, other collaborators, funding sources. I'll be at poster 1 and 28 later if you have more questions or want to talk more. And thank you all.